Alright guys, we're going to go over 3.6, which is cellular respiration. We're going to start with the formula for cellular respiration, which is glucose plus oxygen is going to give us carbon dioxide, water, and ATP. A mnemonic that's going to help y'all remember is go cow. Alright, so we're starting with G, the glucose. This is going to come in our first step, which is glycolysis. You have to remember that this is happening in the cytoplasm, and essentially it's the glucose breaking down by ATP and AT+ and various enzymes. So we start the process out with our 6-carbon molecule, which is glucose. The formula is C6H12O6. And the first step that we're going to see in this is we're going to have 2 ATP molecules with the help of enzymes will come in and start the process of breaking down our glucose molecule. All right, so we are using energy. This is our investment, and so that ATP will turn into ADP, and we end up attaching a phosphate onto each of the 3 carbon molecules that we've broken. All right, so step 1, 2 ATP and enzymes begin breaking down glucose. The second step is where we're going to have enzymes bring in another phosphate and attach it to that molecule we had before, which was called pGal. All right, so now we have 3 carbon molecule with a phosphate on each end. The next step is going to be where our enzymes are going to help NAD+ strip hydrogen and electrons from our molecules. So you'll have NAD+, it'll go in, it's going to strip off a hydrogen, and also it's going to strip off 2 electrons. And we now have a molecule called NADH. I'm going to draw the electrons this time just so we kind of have that visual, and then I'm going to stop drawing those electrons on when we move on. And these NADH molecules will be sent to the mitochondria. Our next step in glycolysis is going to be where we're finally going to make our ATP. We'll have ADP molecules take that last phosphate from each end of our 3-carbon molecule, creating 4 molecules of ATP. And so after this step, what we're going to be left is we're going to have 2 pyruvates, which is what our 3-carbon molecules are, and we'll have 4 ATP. All right, so the formula for our pyruvates, C3H4O3. Now when you're looking at your 4 ATPs, keep in mind that we invested 2 ATP to start out in step 1. So our net gain is 2 ATP. All right, so now we're going to look at the final products that we had. We had 2 NADH molecules we made in step 3. These are going to go on to the mitochondria, and we'll look at these again later. We had 4 ATP that we created in the last step. Keep in mind the net gain is 2 since we invested 2. And we have 2 pyruvates. Now what we are left with, these products that we made from glycolysis, which were your 2 pyruvates, your 2 NADH, these are what are going to continue on to the next stage. Now if we have oxygen present, then we are going to go through the aerobic process, and it's going to move on into the Krebs cycle. If there is not enough oxygen present, we're going to go through the anaerobic process, and it's going to go through fermentation. Let's talk about the fermentation first. So we have 2 types of fermentation. Lactic acid fermentation is the first type. We see this in animals and bacteria, and some examples would be getting sore muscles for us, and we use a bacteria for certain foods like yogurt, pickles, and sourdough bread. The location that this is happening is also cytoplasm because we haven't moved into the mitochondria yet. We're going to start with our pyruvates that we created from glycolysis. And these pyruvates will basically undo what we did in glycolysis. And we're going to take the hydrogen back from the NADH, recreating NAD+. Now that hydrogen is attached to that 3-carbon molecule, that is a lactic acid molecule. It's going to be released as waste, and the NAD+ that we've made are now going to help us restart glycolysis. So keep in mind, when we are going through fermentation, there's 0 ATP in this process because the whole purpose is to restart glycolysis. The second type of fermentation is alcoholic fermentation. This is also in the cytoplasm. Some examples would be with yeast and some plants. Now, we're starting with that same, our 2 pyruvates. Now, the difference that we're going to see is before we take that hydrogen away from NADH, our enzymes will break that pyruvate, releasing some carbon dioxide. Okay, and then when we're left with the 2-carbon molecule, at that point, that's when we're going to take the hydrogen from the NADH. All right, so hydrogen stripped from the NADH, it's attached to the 2-carbon molecule, and that formula of what we have at that point, that 2-carbon molecule with the added hydrogen from the NADH, is an ethanol molecule. It's released as waste, and we then have the 2 NAD+ molecules available to restart glycolysis as needed. So we have to keep in mind that fermentation is the process we go through when we do not have enough oxygen present. It does not produce any ATP, but what it does do is it changes our NADH molecules back into NAD pluses so that we have them there for glycolysis, because NAD plus is essential to complete glycolysis. All right, so now if we do have oxygen present, which was our other reactant in our formula for cellular respiration, we'll go into the Krebs cycle, which is our aerobic process. It is going to occur in our mitochondria. Okay, so more specifically, it's going to occur in the mitochondrial matrix, and it's also known as the citric acid cycle. This is when we have our oxygen present. So look at our reaction, it's that second reactant. So we're starting with those 2 pyruvates that we made in glycolysis, and our first step is going to be where those pyruvates are broken down into acetic acid. To do that, we are going to be releasing some carbon dioxide, and we are going to be releasing some hydrogen electrons to make NADH. So we take that 1 carbon off from each pyruvate, create carbon dioxide, we take NAD+, we strip the hydrogen from the molecule, create NADH, and the molecule we are left with is our acetic acid. So step 2 in the Krebs cycle is where we'll have coenzyme A come in and bond to the acetic acid, creating acetyl-CoA. All right, so we have that 2-carbon molecule, and we add in CoA. You don't really have to know specifically what the formula is. You just need to know we have CoA added, and it turns into acetyl-CoA. Our next step, step 3, this is where that cycle comes in, right? So that acetyl-CoA that we created will bond with a 4-carbon molecule that was made in a previous Krebs cycle. So this purple 4-carbon, this is from the end of step 6, cycling back into step 3. So step 3 is where that CoA is leaving. We have the 2-carbon molecules. We add on that 4-carbon from the end of step 6, creating citric acid, which is the other name. So this is the C6H8O7. You don't need to know the C6H8O7, just know that it's a 6-carbon molecule. Now, step 4, we're going to take that 6 carbon citric acid, break it down into a 5 carbon molecule. And in doing that, we're going to do something similar to what we did in step 1. We're going to release some carbon dioxide. We're going to strip some hydrogen and electrons and make NADH. So take one of our carbons off from the end, make CO2, take some NAD pluses, go in, strip up away a hydrogen, some electrons, create some NADH. All right, so next we'll take that 5 carbon molecule in step 5 and break it down into a 4 carbon molecule. So we're getting closer and closer to that 4 carbon we used earlier. Say we're going to do similar, release some carbon dioxide, make some NADH, but what we are adding into this step is we're actually going to make some ATP this time. So take each carbon off from the end, make CO2, take some NAD pluses, go through, strip some hydrogens, make NADHs. But what's going to be new in this step is that when we are breaking that carbon dioxide off, we at the same time are going to take some ADP, use that energy and bond a phosphate to an ADP, creating 2 molecules of ATP. All right, next step. Now that we have that 4 carbon molecule, we're getting even closer to that molecule that we used in step 3. So now that 4 carbon, we have a 4 carbon, but it just needs to be rearranged a little. And that rearranging is in the form of stripping away some hydrogens and electrons. So we'll take the 4 carbon molecule we're left with at the end of step 5. We will strip away some hydrogen, making NADH. And the new thing coming in is we'll have FAD go in and strip away 2 hydrogens, creating FADH. So something a little different about FADH2 is that it's only going to hold 1 electron, whereas as we remember, NADH carries 2 electrons. So I want to draw that in on this part just so we remember. Then from step 6, this is the 4 carbon molecule that's going back into step 3. Let's look at our products. Carbon dioxide, CO2, we built it in step 1. You see the 2 molecules there. We built some in step 4, some step 5. Altogether there's 6 of them. NADH, we built some in step 1, step 4, 5, and 6. As you can see, we have 8. FADH2 was only made in step 6. We have 2 of them, so there were 2 FADH2. We only made ATP in step 5. We made 2 of them, so there are 2 ATP molecules built. If we come back to our reaction for cellular respiration, you can see the carbon dioxide, which is one of the products. We are getting it in Krebs cycle. We are making 6 carbon dioxides in Krebs. Now the rest of this, the NADH and the FADH2, are actually going to go into the electron transport chain, which is our next step. So electron transport chain, the last part that we are going to look at. This is not going to be in the mitochondrial matrix. This is actually mainly going to be in the inner mitochondrial membrane. So we're going to be looking at our protein complexes in the membrane. Now within the matrix, we will have the NADH and the FADH2 that was created in Krebs cycle, and remember that NADH was both glycolysis and Krebs cycle. So the first step is going to be where our NADH and our FADH2 are delivering hydrogen ions and electrons to our protein complexes. So hydrogen ions are going to get into place at the end of the protein complexes. The electrons are going to go into that membrane, and the next thing we're going to have is that those electrons, they're going to activate those protein channels so that those protein channels can then pump that hydrogen that's now in place out. All right, so you can see your electrons will go through, activating those pumps. And when they go through, activate the pumps, those pumps will then pump those hydrogens that are in place out of our matrix. Now those hydrogens that were transported out are going to start to accumulate in a large amount outside our mitochondria. Now that large accumulation is going to lead to a rapid diffusion, because remember that our substance is going to go from high concentration to low concentration. So now that we have a high concentration outside, they're going to diffuse back in to the area that has a lower concentration. That brings us to our next step. So those hydrogen ions that are going to be that are going to diffuse are going to activate ATP synthase. All right, so they're going to go through, and the mechanical energy from them moving through the ATP synthesis and like turning that wheel is going to be the mechanical energy that can then be used to take an ADP molecule and bond it to another phosphate, creating ATP. So the mechanical forces that are activated by that diffusion are bonding ADPs to phosphates, creating ATP. Now this is the step that's creating the large amount of ATP, because it's going to repeat up to 34 times. All right, so this is our ATP synthase. This is where the ATP is being created. Last thing is cleanup. Now that that hydrogen ion has diffused back into the matrix, we've got to clean it up. Okay, so this will be cleaned out by being converted into our last product that we haven't talked about yet, which is H2O. So the oxygen from the air that you breathe, along with 2 of the hydrogen ions that have diffused back in, 2 electrons that have gone through and activated the protein complexes, will bond, creating H2O, which is our last product, one of our last products. ATP, let's summarize. We created a net gain of 2 from glycolysis. We made 2 in Krebs, and we're going to make up to 34 in the ETC electron transport chain, giving us a total of up to 38 ATP. Now, there are different ways that this is looked at. Some places are going to count ATP differently depending on looking at the NADH and FADH molecules that are being made in glycolysis, because those are what lead to the ATP being made in the electron transport chain. But I'm just looking at it in terms of counting the exact ATP made in each step. And so this would be our overview of what we have all together through the process. So this was our quick summary of cellular respiration, going through all of our steps and looking at how everything is made and where in the process we have everything made.